It's a seminar series, a Ph.D. scholar series. So we have Sowmini Shankara here, who did her undergrad from the Kakatiya University in 2001 with a gold medal in chemistry and university rank of 6th. In 2003, she obtained her master's degree in biotechnology from Osmani University. She joined the genetic transformation lab at ICRISAT in October 2005 for her MTech dissertation work, and she obtained her MTech degree in biotechnology from JNTU, Hyderabad in 2007. Subsequently, she registered for her Ph.D. degree in biotechnology at JNTU in October 2007, and she was a recipient of Junior Research Fellowship as well as Senior Research Fellowship from the University Grants Commission in Engineering and Technology scheme, and from October 2009, mainly for her PhD work. She has 3 posters and 1 oral presentation in international conference and has 2 publications to her credit. Today she will be presenting her research work on this title, Development of Groundnut Transgenics for Resistance to Aspergillus flavus. So I assume she'll take about 35-40 minutes at the most, and then we'll have question and answers from you. Okay, so Sowmini.
Good morning to everyone. Thank you very much, sir, for your kind introduction. Today I'm going to speak about my thesis topic, that is development of groundnut transgenics for resistance to A flavors. Groundnut being an economically important oil and protein-rich crop, it is a seed-propagating and self-pollinating legume crop. It is a very good source of high-quality edible oil and carbohydrates. It has easily digestible protein. It is a very good source of animal feed and fodder. Various uses of groundnut. It is used in the form of fuel, soil conditioners, peanut butter, fodder, chemicals, cork substitute, and for cardboard. The main constraint for production, coming to area and production, peanuts rank the 3rd in position among oilseeds. Developing countries contribute about 97% of world groundnut area, which is mostly grown by smallholder farmers in subtropical and tropical regions. India being the largest peanut growing area with 4.9 million hectares and stands 2nd in production with 5.78 million tons. It has an average yield of 11,794 hectogram per hectare. The main constraint of groundnut is the infection with Aspergillus flavus. Groundnut is the most susceptible crop. These fungi are weak facultative pathogens and can exist as saprophytes growing on organic nutrient sources. These fungi are ubiquitous. They reduce the agricultural yield and decrease the quality of harvested grains, and also makes the produce unfit for consumption. Aflatoxins are the toxic secondary metabolites secreted by infection with A flavus and Aspergillus parasiticus. These toxins are associated with both acute and chronic toxicity. They are carcinogenic, causing liver cirrhosis, thereby liver damage. These are teratogenic agents causing tumors. They also suppress the immunological responses. Several countries have imposed stringent limits on the levels of aflatoxins permissible in food products, such as USDA has limit of 20 microgram per kg and European Union has 2 to 4 microgram per kg. Where host cells die within few hours of pathogen attacks, thus thereby limiting the availability of nutrients for the further localization of the pathogen. There are 3 reasons of pathogen failure to cause a disease. Either they are unable to support the nutritional requirements of the pathogen, or they have some barriers, structural barriers or toxic compounds which prevent infections, or they show the defense mechanisms on pathogen attack. These plants express a wide variety of genes called PR genes when there is a pathogen and pest infection. In peanut and A flavus interactions, there is number of genes getting activated, such as chitinases and glucanases, which hydrolyze fungal cell wall. Also, there is an allelic active oxygen burst, which also helps in playing an antifungal role. There is also an elevated allox activity, thereby increasing the membrane lipid content, thereby causing cell deterioration and hypersensitive death. Hence, there is a need of developing allied cultivar with the inborn resistance. Developing an allied cultivar with the desired trait is an integrated approach of technologies like breeding, genomics, transgenics, and white hybridization. Breeding plays an important role in screening the germplasm and constructing SSH libraries, SAGE libraries, etc. These libraries can be used as a source for selecting candidate genes which are either used by genomics technology as for marker-assisted selection or by transgenics using gene cloning and genetic engineering techniques. For functional analysis, either by gene silencing, overexpression, or telling. White hybridization also plays an important role in developing allied cultivars. Here at ICRISAT, we follow breeding, genomics, and biotechnical genetic engineering approaches for combating A flavus constraint. In our department, in our lab, we are working on 3 genes. These are chitinases, differences, and lipoxygenase to address this issue. Depending on the source of gene, there are 2 approaches for fungal resistance, either pathogen-derived resistance or non-pathogen-derived resistance. Pathogen-derived resistance is the resistance where a complete gene or a part of gene is introduced in the plant which subsequently interferes with one or more life steps in the life cycle of the fungus. Non-pathogen-derived resistance, on the other hand, helps in utilizing host-resistant genes for adaptive host processes in response to pathogen attack. As lipoxygenase are hypothesized to play an important role in mediating plant-pathogen interactions, lipoxygenase catalyzes the oxidation of polyunsaturated fatty acids such as linoleic acid and linolenic acids to produce unsaturated fatty acids. LOX genes increase the resistance through jasmonic acid, which is reported to be a potent inhibitor of aflatoxin biosynthesis. Jasmonic acid is produced in the pathway to allene oxide synthase pathway. In vitro evidence has showed 13S LOX as a putative resistance factors. Hence, we have taken genes PN-LOX2 and PN-LOX3 being 13S LOX producers. There is also reports that exogenous 9S LOX application extended the time of aflatoxin transcription, but 13S LOX has inhibited the aflatoxin gene transcription. Hence, we took 13S LOX genes, so that is PN-LOX3, for our transformation studies. With this, my objectives of the present study are subcloning of the gene, which we, like peanut lipoxygenase gene, which we got from Dr. Nancy Keller from University of Wisconsin, into a marker-free binary vector under the control of constitutive 35 promoter. Then transformation of downer genotypes using this binary vector using Agrobacterium tumefaciens method, identification of transgenic plants showing high expression through PCR, RT-PCR, and southern blotting, and finally confirmation of resistance through fungal assays and ELISA. The gene has been subcloned by following various steps into marker-free binary vector under the control of 35 promoter. This construct has been confirmed by different restriction enzymes. We move forward with Tropics transformation for preliminary check whether the gene is expressing at RNA level, and we could confirm that the presence of transgene by using internal primers amplifying 1300 base pairs. Then we move forward with Groundnut Transformation protocol, which we follow in our lab, developed by Dr. Sharma and Anjaya. I have used GL24 genotype since it's being a short duration and is susceptible to Aspergillus flavus. Here we used D-split, D-coated, D-split, de-embryonated cotyledons, which were transformed using Agrobacterium tumefaciens method. We have confirmed the presence of transgenes using PCR and RT-PCR analysis. PCR was carried by junction primers, wherein we amplify a fragment of 714 base pairs, which was designed between the 35 promoter and the gene of interest. and RT-PCR was done by using internal gene primers, amplifying 1300 base pair product. Similarly, PCR analysis and RT-PCR analysis was carried out in till T1, T2, T3 and T4 generations. The presence of transgene the integration of transgene was confirmed by performing southern blotting analysis of T3 transgenics carrying the binary construct, by performing Him3 restriction enzyme analysis to check the copy number. And the inheritance pattern of the peanut lipoxygenase gene was performed by performing chi-square analysis, wherein almost all transgenics are being following the Mendelian ratio. Similarly, inheritance pattern was checked in T1 and T2 generations also. Coming to the final objective, performing a fungal bioassay, we followed a protocol where we could separate the root and pod zone using a new technique where the seed is planted in the tube so that the root goes into the pod and the pods are separated on overlay. And drought was imposed to check A flavors and drought interactions at 75 days of sowing. Inoculum used was sorghum seed infected with A flavors AF11-4 obtained from empty well lab. Inoculations were done at 5 levels before sowing, 3rd week, 5th and 7th and 9th week after sowing. Drought imposition was done after 75 days of sowing, and root zone was watered until harvest. Post-harvest, screening techniques were performed to check post-harvest in vitro seed colonization, wherein we maintained 30% seed moisture using a plate technique, which was incubated for 7 days at 28 degrees. The presence of Aspergillus flavus in the soil samples was regularly checked by collecting soil samples at sowing, flowering, harvest stages while performing the experiment, and these were petted on AFPN media, which is selected media for the Aspergillus flavus and parasiticus, and the colony forming units were recorded. Later, ELISA analysis were carried out to determine the amount of toxin present in the samples by following indirect competitive ELISA. From these results, we could report that more than 90% of the positive transgenics screened under glasshouse conditions contained aflatoxin levels within the limit, that is below 20 microgram per kg. GenStat analysis was carried out to see whether there is a significant difference between controls, non-transformed and transformed events. Though we could not find any difference in percent visual infection, there was difference in the aflatoxin content, thereby explaining that different mechanisms are involved both in A flavus infection and aflatoxin. Both are. Later on, we confirmed the protein expression studies by performing a page analysis of the bacterial induced protein. We also performed the pepsin digestibility studies to check whether the protein is getting digested at 37 degrees in presence of simulated gastric fluid containing 0.3% pepsin. And sampling was taken at different time intervals ranging from 5 seconds to 2 hours. And we could find the digestibility of the protein starting from 5 seconds. I was fortunate enough to be involved in the isolation of seed-specific promoters from legumes, wherein we have isolated 2 promoters, chickpea lectin promoter and groundnut seed-specific promoter. Sequence analysis was done and motif search was done using various bioinformatics softwares. These sequences are reported in GenBank. Further promoter activity confirmation was done through EMSA studies, that is electromobility shift assay studies, wherein we have done the binding assay reactions in the presence of nuclear proteins with different plant parts, this leaf, seed, root, etc. And we found there is an interaction between seed nuclear proteins and the PCR amplified probe. Even with the chickpea lectin promoter, we have observed a shift in the band when it is reacted with nuclear proteins of seed. Later on, we move forward in constructing binary vectors with these promoters to confirm the expression of GUS gene, and we have done the transformation of Arabidopsis and tobacco for preliminary checks. Arabidopsis transformation was done by modified floral dip, where it showed seed-specific expression in Arabidopsis, and later on tobacco transformation was done using leaf disc transformation. This also showed seed-specific expression. The presence of GUS gene was also tested in using GUS gene primers in T0 and T1 generation tobacco transgenics. In summary, I would like to conclude my presentation. We have subcloned the binary vector, subcloned the LOX gene under constitutive and tissue-specific promoters, developed 25 marker-free groundnut transgenics carrying peanut lipoxygenase gene. We have confirmed the presence and integration of transgene over generations, that is till T4, by PCR, RT-PCR, and southern blotting. We have used a new technique of screening for fungal bioassays under greenhouse conditions to evaluate transgenics and to understand A flavus drought interactions. And we have isolated 2 novel seed-specific promoters from groundnut and chickpea. Future prospects are production of transgenics with lipoxygenase gene under the control of chickpea lectin promoter and groundnut seed-specific promoter, and molecular characterization of these transgenics, purification of LOX protein from PET-induced bacterial culture, and standardization of LOX enzyme assay so that it can help in checking the transgenics also, and real-time PCR analysis of these transgenics for copy number and quantitative expression studies is to be done. I would like to take this opportunity to thank Dr. Sharma, who has been the mentor all through these years, and Dr. Farid Walia, who is my co-supervisor, and special thanks to Dr. Pooja, who took care of day-to-day experiments. I also like to thank the entire scientific, administrative, and supporting staff of Genetic Transformation Laboratory and Mycotoxin Laboratory. In addition, I would like to thank LSU, FETS, FMU, Security, and Library for their services provided. I also would like to thank the funding source, UGC, for giving JRF and SRF, and finally my family, who will always be my entire support. Thank you.
Thank you, Sowmini. Are there any questions for Sowmini?
In one of your slides, you mentioned post-harvest seed colonization. I think that is wrong. That is pre-harvest seed infection. That is post-harvest seed colonization. I think that is pre-harvest seed infection, right? After harvesting, you check the seed whether infection is there or not, right?
Yes.
And that is when we come to post-harvest. Pre-harvest seed infection percentage.
Pre-harvest means?
I mean, whether the infection is there. You have applied.
So you picked up whatever is coming.
You applied some inoculum, right, during pre-harvest conditions. So once you harvest, you need to check is there any infection is there or not.
Or is it in vitro seed colonization?
What did you do?
That is different. We inoculate with the spores.
So you picked up the seed and then just cultured it.
Yes, we didn't inoculate anything.
Yeah.
We just checked whether it's one.
Then this is post pre-harvest.
Pre-harvest.
Anything else? Any clarifications?
So I think the main problem is there in the seed. Then usually I use the seed specific
For that reason only we have cloned the gene under the seed specific promoters also, but the development of transgenics is under progress.
Any questions? Clarifications? Just want to check up, what do you mean by Mendelian inheritance? Can you go back to this? Just explain this.
I have done just the chi-square analysis, sir.
Yeah, that's good.
Whether it is following the regular inheritance pattern or
Can
not.
you go back to this slide? So, why did you do Mendelian inheritance?
to check if it is not behaving apart from the
So you
transform.
are assuming it's a single gene inheritance?
Because we have cloned only single gene.
No, no, but to copy numbers, or how do you account for copy numbers?
For that,
So this is the assumption that these are all single copy inserts?
yes, sir.
Hmm?
Yes, I am assuming that.
And you follow it, get a 3:1 ratio, or
Mostly, sir. Not every
maybe you didn't get it.
1-1-3 here. Even in T0 we didn't get.
And what is the purpose of pepsin digestibility? Why,
To check whether the protein gets digested even if taken in.
so why, how is it related to this work?
It's like related. It's a transgenic protein which we have to answer the questions of biosafety thing for that.
So you should mention that. Because otherwise it doesn't make a connection. Why are you doing protein digestibility? You should mention that you did it because of the biosafety considerations. Make sure that this protein with which you're going to work in the future is safe, at least as far as pepsin digestibility is concerned.
Sowmini, I have a question. This aflatoxin content in 2 levels that you have shown, is it the greenhouse experiment or the field one? So you didn't mention the field data? It would be good to add both of them and see, they're the same events, right?
Yeah.
So it would be good to see how it happened in the microscope process.
You should have included the data, because that's a part of the work. Why did you skip it?
That's not good.
Are you not convinced with that data or what?
Actually, some part of experiment should be done with that also, sir. So it's not concluding. We have to check with one more experiment. We didn't get the aflatoxin then. We
The range. Doesn't matter.
No, not the range. To test the plating samples again, to completely analyze the data, we didn't perform that experiment. That's why they moved. Crash.
So are there any suggestions for Sowmini to further improve it? Because after this seminar she is supposed to actually write and submit. So one of the purpose of a seminar like this is also she should get some guidance on what she missed out, or can she do something better, or that will help her in actually writing up the thesis. So if there are any suggestions, will be very welcome. Yeah.
It's not the technical aspect, because I really don't have something like that. But there was one section in your summary where you said you developed a novel strategy. Was this something as a result of your study, or did I get it right that this is something new that you developed?
We have used, that was developed in the pathology laboratory, and we have used for screening our transgenics.
So the novelty is the first time it was used for screening transgenics.
That's all, okay.
The pathologists I think use it on a routine basis, but a lot of the screening happen in the field. The challenge here is how to use such techniques under confined conditions. So that are you ready?
No, actually we have done pathology only once, and after that this is our second trial. Just we tested the system, we have developed the system, and we have not done anything in pathology much, just we did only one experiment.
Yeah, because it's very important to have a good phenotyping system. Otherwise, you can produce many transgenic plants, but they will not be, you cannot do good screening. And if you don't do good screening, then your transgenics events which you produce are no good then. So I think in this science always there is a challenge of how best you can screen it under confinement, because you don't have the luxury of taking the transgenic material out into the field and doing it under natural pressure. So some of the challenges are how best you can create a disease, how best you can get a disease reaction and the host-pathogen interaction, and move beyond from the greenhouse to the field based on whatever you can select under field conditions.
You didn't include your list of publications. So at least for the benefit of the audience here, you can also tell us what is the status of the publications from the work that you have been doing?
I have one publication to my credit, which is on groundnut seed promoter, and one is under submission. I have presented in conferences, oral and poster presentations from the work what I have done.
But would be nice if you can submit that, because we're also keeping track of this document.
Yes, sure.
Actually, now your transgenics are in progress. I think you are using the seed-specific promoter. Does it have some jasmonate-specific elements? Because the jasmonate signaling is very important for the
Yeah, we haven't checked that level. That's what we are looking forward to isolate the proteins, bacterially induced proteins, and then validate the results. For the enzymatic assay also we need to perform.
I have one suggestion for me. Maybe for your thesis, you can add a table instead of using percentage. You can have a table with a percentage of transformation events and how many you have screened and how many have finally gone to the field, something like that.
That's been the regular earlier thesis.
Like everyone as real summary of your work.
Because it gives it at a glance, how much effort was involved, how many events you produced, how many others turned out to be positive. I think it's good to give that consolidated result for anyone reading the thesis to get an idea how did you go about doing it, what was the generation of that material.
And also some of the challenges that you have encountered with your material, because that's very important.
Yeah, because everything looks very rosy here, but I'm sure it's not that simple.
Yeah, it took her long to really
Many extensions.
She has spent a long time.
So I think you should have mentioned, or so that if someone else is also working in that area,
Exactly.
so they'll have some.
Yeah. For example, you're talking about the LOX genes, and that itself was a challenge because the LOX genes, they're involved in the oxalate pathway, so they're likely to interfere with the lipid metabolism as well. So that is one of the reasons why we went into promoter. Otherwise, there's no link of promoter to this work. But the very fact, there's no other way of doing it, using a constitutive promoter. So that's why the idea is to actually identify seed-specific promoters so that the whole plant is not affected. Because many times when you're doing metabolic type of engineering, there can be adverse effect on morphology of the plants as well. So I think you should highlight when talking of these things so that the audience understands why did you work so long, plus why did you go for things like promoter cloning, Otherwise there's a disconnect between promoter work and anything else? If not, then let's thank Sowmini for the presentation as well as wish her all the best for her thesis.